privilege to pick the speaker every year for this. And I tend to pick people that I know well. In fact, I always pick people that I know well because I have great respect for every one of our speakers. You remember last year we had Linda Giudice in a similar manner, giving you your talk virtually. And it was very well received. And this year we have Dr. Richard Scott, who's joining us, I believe, from South Carolina. Although Richard's kind of like, where's Waldo? He could be anywhere in the world. He really is a world traveler and speaks all over the world on these topics related to assisted reproduction. I think I've known Richard now for maybe 35 years. I mean, it seems like a long, long time. I first met Richard, I believe, in a meeting where he was presenting when he was a fellow at the Jones Institute in Eastern Virginia. Again, one of the best places to train, especially back in the 80s when the Joneses were still alive and still doing the best research in the field of assisted reproduction. It was the place to train in those days. He was in full Air Force uniform, as I remember, because he was in the Air Force at Wilford Hall doing his residency and then fellowship in Eastern Virginia. He stayed in the military for, I believe, another 5 or 6 years in Bethesda and Lackland, part of that service, of course. And then he came to New Jersey and was the real founder, true founder of RMA New Jersey. He has left that position. He's been a member of this department and chief of this division since 2006, I believe, for a long time. And he has been a true supporter and done so much for this school without really even having to ask, including a fellowship, training the residents and our students. And so I'm very proud of that relationship with him. He's been a true friend of this department. He's been a true friend to me. I really value his counsel. You know, there are people that you go to when you have real questions and concerns. We all do. And Richard's one of those guys. So I'm really pleased that he's joining us today as still a member of our voluntary faculty. And so I hope you enjoy his talk, and I'll turn it back over to Gary.
All right, thank you so much. Okay, Dr. Scott, whenever you're ready to begin.
Well, great. I also want to thank everyone for being here as I transition my role within the department. Obviously, having left RMA and in New Jersey, but I do get back from time to time, and I will always be available if there's ever anything I can do.
Dr. Scott, I'm so sorry. We are suddenly not hearing you.
Uh-oh.
Michelle, can you hear him?
Yes, Dr. Eber, I can hear him.
Okay. I stopped sharing. I'm not muted. Can anybody hear me?
I hear you, Dr.
Okay.
Scott. Yeah.
Sorry about that. Now we hear you loud and clear. Go ahead.
Okay. So make sure, and can everybody see my slides still?
Yes, we can.
Okay, just want to make sure. So I've been part of the department for a long time and consider it my academic home. And I will always be available to some of the people that I've known there since from the very beginning. And I have strong connections to lots of your faculty. So it's exciting to be here today for this important day. Let's now stop sharing. Well,
I still see your slide, Dr. Scott.
it's not allowing me to advance.
Okay. You want to click Start Slideshow. I think that might be the better option. If you go to Slideshow at the top,
Okay, well,
and then you'll click all the way to the left from beginning.
yeah, okay. I'll try again. There it goes. Okay. I have no disclosures. And I do work for a foundation that used to provide the service clinically, but they don't anymore, and therefore I have no potential conflicts. This is a slide of Louise Brown that was taken on her 40th birthday. Many of you may have seen this picture. It's been
Dr. Scott?
Yep.
I'm so sorry to interrupt you. The slide that you are sharing is a different slide. There you go.
Okay. Hmm.
So what I think you can do is, do you see the full-size presentation on your screen, or do you see the editor's plane?
I see the full-size presentation.
Okay, so if you could go to the share button, there's usually a little triangle next to it. And if you click it, you'll have the option share content. And then if you click share content, you can choose what you want to share, and you can share the full size slide.
Okay. I'm going to stop sharing for the moment. Hold on. I'm so sorry, everybody.
It's not a problem. It's no big deal.
A little bit. So there's the window I want. That's the screen I want. I don't see a whole lot of other options. Can you see
Okay,
it now?
we see you and hear you. We see the editor's plane, which is okay. Can you go to the right-hand side next to the baby and just hit, that's fine too. Go ahead.
Does it work now?
So we still see the editor's plane. Can you find the plane where you would be able to edit or sort your slides and just click the down arrow there? No, on the right-hand side, all the way over on the right.
Okay.
And then just there, as though you're editing your slide, can you just click down?
Well, that would be down.
Yep, that's good. Now we see the next slide. So we can just do it like that, and that will work fine.
Okay, then I will do that. Thank you, Gary.
No problem.
My apologies to everybody.
It's no problem.
Like Dr. Sauer, like a lot of your faculty, I've done a few talks. You think I'd know how to do this, but I apologize for the challenges. That's Louise Brown on her 40th birthday, and that was her incubator. Can you imagine? That's what technology looked like when this started. She was attempt number 128, although there have been different versions of how many attempts there were, but Dr. Edwards once told me it was 128, so I'll believe that. And on the right is Elizabeth Carr, who was the first baby in the U.S. It was from a U.S. program, which came, of course, almost 4 years later, 3 years for the conception. So the reality is things have changed a lot since then, but we still weren't very successful. And so we've been trying to understand for a long time what's been impairing these embryos. They look normal. They go back at the right time. The uteruses, the endometria look normal. And so the last 20 years of the work in this field have focused principally on trying to understand why these morphologically normal embryos sometimes don't make babies, which of course burdens our patients with failure and more uncertainty, and is just is an incredible burden to them. If you look back at the biology, you get some of the answers. This is a classic paper. For those of you, it's not just for REIs, written by Terry Hesselt and Pat Hunt, who are now they're just about to retire from Washington State. They were at Cleveland at the time. But a Nature Review Genetics paper that talked about the fact that aneuploidy risk, when they looked in clinical losses and ongoing pregnancies, but principally clinical losses, there was a little J pattern where it was a little higher when you're in your very early teens, but then it's nadir in your teens, and then it increases linearly up to your early 30s, and then more rapidly to about 38, and then exceedingly rapidly after that. But the prevalences were still relatively low. Even at 42, it was a third of pregnancies, but obviously 2/3 would have then been euploid. This is a paper from Chaim Jallis. It replicates work done by Jason Fernasiak when he was a fellow at Rutgers as part of that program. This shows that the prevalence in embryos rises much more rapidly. Most embryos that are chromosomally abnormal just don't implant, as you know. And so by the time you get to your early to mid 40s, it's over 90%, 95% almost of embryos are chromosomally aberrant. So really looking at embryos is just a continuation of what's been going on in our field now for 60, 70 years. In the 50s, when babies were born, we looked to see if they looked like they had Down syndrome or anything else. That's how it was screened for. Not the most sophisticated approach, but all that was available. Amnios began in the late 50s and 60s. They became ultrasound guided on a fairly routine basis by the late 70s. CVSs came around in the early to mid 80s, quad screens and nucleolucity scores. And then in the 90s, we started screening embryos, all part of this effort to try to identify aneuploidy earlier and to make for safe and healthy babies, safe gestations and healthy babies. FISH was used initially because it didn't require metaphase cells, which was a big advantage, and it was completely inadequate. FISH was a disaster. If you look at this particular slide, you'll see that looks almost like there's 3 red dots. Over here, there's 2 of those dots. That's hard to tell whether it's 1 or 2 dots because of the potential for overlapping signals. But in fact, that came from an embryo that was euploid. So many technical problems. It was simply not up to the task. This was followed by microarray. And if you look at this slide, these are the dots, 262,000 dots. They're actually there. It's not a graphic representation. That's the actual scattergram. And you actually have to squint pretty hard to see that the 8th chromosome, it's represented by this column, is actually slightly higher than the other ones. Look how close that is and how much of the data falls in the normal range. Now, with a bunch of fancy statistics, and we did that right here at our program, we were able to show statistically that was probably a trisomy 8, maybe with 98 or 99% accuracy. But boy, there was a lot of scatter there, a lot of overdiagnosis, a lot of underdiagnosis. While it was dramatically better than FISH and dramatically better than CGH or array CGH, that had also come along by then, the reality is we overdiagnosed 4 or 5% of the time. This was followed by qPCR, which looked at only places where we could be more accurate, but we only looked at 4 places on each chromosome. This would be an example of a monosomy 22. But look, some alleles dropped out or are overrepresented. This is a big problem. All of this has been followed up, and I promise to be to the point shortly, by using NextGen, and it can be done with different kinds of amplification. I will not burden you with those discussions, although they're interesting. And this is the trisomy 13, where we see a copy number analysis with clear separation. We also can look at the SNP profile to see whether it's the major allele, the minor allele, or a mix of the two, which confirms when you see this kind of those 4 clusters that is in fact a trisomic chromosome. So we have both a qualitative and a quantitative assessment of what's going on in that cell, and that's been done. And the benefit, while some would debate it, those who really know the literature, I don't think would. Implantation rates have gone up from the low 40s to the high 60s. Ashley Teagues, I'll show you some of her data in a minute, did a non-selection study using this technology. And it clearly shows the power of the predictive value of the test. However, when we apply it clinically, while we do see benefit, it's empowered elective single embryo transfer on a consistent basis. It's gotten rid of almost all the multiples, all the polyzygotic multiples. It's done many positive things, but the reality is you're only kicking up pregnancy rates about 20%. And so we still have a 31% failure rate. We're still failing too often. Of course, this is much better than when Dr. Sauer and I were fellows many years ago, when implantation rates were in the 4 and 5% range, at least in Norfolk they were. We put back 4, and so we were getting cumulative rates in the mid-teens. But now with 1 embryo, we can get to about 2 out of 3, but we still fail far too often. And so the challenge has been to try to explain why you have a temporally appropriate, morphologically normal or even ideal embryo, clear intercell mass, beautiful trophectoderm. Why does it often, a third of the time, fail to implant and progress to delivery? And so I thought I'd share with you some of the things that we've been working on today to try to answer this question, to try to take embryo diagnostics to the next level. This is a slide of kind of the summary of what we've been able to learn about genomic instability in embryos. While we're very focused on whole chromosomal aneuploidy, it's the most important problem. This is a monosomy 16 up here at the top on the right. The reality is we see other types of problems like a mosaicism, as in this mosaic of 10, or segmental abnormalities. And those are really interesting. It's where there's a deletion or a duplication, in this case, a duplication of a segment of one chromosome. So it's not the entire chromosome that's been that has a low copy number. It's part of it. So there's specifically a duplication or deletion. And in this very cartoonish, this would be chromosome 6 here, that has a deletion, a partial deletion. A deletion just means it's on one copy of one chromosome has a copy and the other one doesn't. Duplication means that one has 2 and one has 1, normal, obviously 1 apiece. And so the reality is that when we're able to see these things now, because the analytical techniques we have available to us have the resolution to discriminate them, whereas all those ones I showed you before simply did not. This is actually Teague's study. She did that while she was a fellow right here in this program. And it was a very interesting and important paper. FNS selected her as Investigator of the Year. A lot of interesting things happened because of this manuscript. And it showed some important things. One, it showed that euploids implant about 2/3 of the time. We call it aneuploid. It doesn't implant. 0 out of now almost a little over 450. In fact, it's up to almost 500 now. 0 of those ones that were called aneuploid were actually implanted and progressed to delivery. Mosaics had essentially normal. People get very nervous over mosaicism. Overall, in the way we use them practically, it may be slightly lower, maybe about 60%. But when you're unselected by morphology, they do just as well. But look at the segmentals. Only half as many of those embryos. And these were never detected before. We call them normal. And they certainly are in a position to explain some of the failures. The problem that we've had clinically is that these tests, when they were developed to detect abnormalities of the whole chromosome, an extra copy number, a trisomy of the whole chromosome, or missing an entire copy, monosomy of the whole chromosome. And if you look at the assay that we developed over in Basking Ridge, you can see that we don't do whole genome amplification. It's extremely unreliable. We do targeted amplification. But look, we don't test everywhere. That means we're only testing some of the sites. And you can see, for instance, on the first 4 megabases of chromosome 1, we don't test at all. On chromosome 2, the outer 8 megabases. Look at chromosome 1 in the centromeric region. It's huge. You'll see many gaps throughout these chromosomes, even on the smaller chromosomes. 19 is a disaster. So the reality is that when we see a segmental abnormality, it's bad, but we don't really we've not had an analytical assay that was designed to enhance the detection of them. So it's possible, and that was our question, that we were missing some of these abnormalities. So we had, in fact, chromosomally abnormal. We had genomic imbalance in the embryo, but our contemporary techniques were not detecting it. This was also a big problem because you need at least 5 consecutive SNPs or alleles to be abnormal, show a gain or a loss at the same level to make the diagnosis. So again, on chromosome 1, the first copy was at 4 megabases, just as an example. But to get 5, you had to go 12 megabases in. These are huge deletions. And the deletions do tend to aggregate in the telomeric region, as we'll see. So these truths are held throughout the chromosome, of course. It doesn't just have to be at the telomeric end. You need a lot of them. And that's what an enhanced assay would potentially be able to provide. So that's what we tried to do. And in fact, is what we did. So if this is what I showed you before, this has about 4,400 amplicons spread throughout the genome. They're pretty evenly spread, but not perfectly spread as I showed you. All the chromosomes are pretty adequately represented, and the technique has been very valuable. But we went back and we said, let's not do 4,400. Let's do 66,000. This took about 3 years of development just to get the primer mix right, because you're doing PCR at 66,000 different amplicons at the same time. And that can cause problems with vastly uneven amplification, all kinds of dropout or preferential amplification. So the reality is it took a long time to find the right amplicons and to get them to uniformly work. This graph is without any statistical smoothing. So this is the equivalent of that green dot when I showed you on the SNP array. You can see that there's some scatter, but there's also, they really center very nicely around what's on the log2 ratio plot. 0 would be 2 copies. And so you can see that's a 46XX. And there's a lot of data point. When you have thousands and thousands of data points on each chromosome, then your statistics become very powerful and you get very certain about copy numbers, even for segments of those chromosomes. And sometimes, indeed, we do see segmental abnormalities. This one has a deletion out here on the 1 and another one on 4, and is actually mosaic for chromosome 14. This is a pretty messed up embryo. Most are not like this. But I thought I would just show you that when you see a deletion, in this case, it's true for duplications as well, it's not that hard to identify that there's a shift in the mean right here to a different mean down here. And the statistics to do that is actually pretty simple. And you can see there's a whole shift of the mean here, just not all the way down to a copy of 1. That is, in fact, a mosaic of 14. And so when we do that, we went back and said, how often does this happen? And we were able to take samples from embryos that had been screened under the old qPCR assay, where we knew the clinical outcomes, but it had no next-gen. So there was no potential for a segmental or a mosaic diagnosis to alter the test. And we went back and pulled thousands of those out of the freezer. And in fact, we looked at 2,379. This requires just an extraordinary amount of time to go back and reamplify and do all those analyses. The enhanced assay requires a lot more data. The little flow cells that we can do 24 embryos on one flow cell at one time on the contemporary technique, PGTC, we can only do 2 of these because there's so many more amplicons. And so this took many runs and many adjustments to the platform, a lot of work. Anyway, that was fun. What we found is that 17% of the embryos had a segmental abnormality. Now that's compared to 6 to 8% that we find across the board in our cross-sectional population using the old assay. So I think it provides great statistical power and great clinical confidence to say that the number of segmentals out there is at least, because even this assay is not perfect, it's at least twice as high as we thought it was. Now that raised the question, and for those of you who are thinking clinically, and I hope you are, you said, well, that's great. There are twice as many, but do they matter? And so there's the prevalence for the enhanced assay, about 7% or so in the current assay. And so we went back and started looking at the nature of these things to try to see what we could learn about the biology and their clinical impact. What we found is that there was a higher prevalence, much higher, on different embryos. Now, this is a percentage of the embryo and the percentage of the displacement. Without going into all the math, there's a higher prevalence on some chromosomes and lower on others. And even if you adjust for the number of base pairs on the chromosome, there are higher numbers on some chromosomes. They are not more likely to occur within a specific gene. It's just whole segments of chromosomes. It's not just at hotspots or crossing over spots. And so we have a lot to learn about what predisposes to these abnormalities. The type of changes that go on during somatic cell division later in life, mitotic divisions, are not responsible for these errors. The mechanism is different. And so, again, there's a lot of work remaining to be done there. I won't bore you with the biology, although please feel free to ask. We found that these were almost always telomeric in nature. It's not true of the ones we find later in life and in people, where they can be intragenic, intrachromosomal, or most often centromeric. If you look at ours, we had relatively few centromeric deletions, a few that were intrachromosomal, and then really almost everything out there was telomeric. Interestingly, we found that there was no relationship with age. So it was 17% of embryos, and it didn't really make any difference how old you were. So I know it sounds mean, it isn't intended to be inappropriate, but for us, that's a relief, because now we have something that can explain some of our failures, even our very youngest patients, right? Because if you're 30 and you have a chromosomally normal embryo, it goes back into a normal endometrium at the right time, it's synchronous, and it's a PGTA normal, really should make a baby. This is one reason that it may not make a baby. So we're gaining insight that could be useful potentially clinically. It does not at all correlate with morphology. So you can't look at the embryo and say, hey, this one's going to be more likely to have a segmental or not. That's why you have to screen. For those of you who are interested about when this might impact development, it's very interesting to see that while the loss risk was slightly higher, the reality is that most of the diminution in outcomes was evident, the difference between euploids and segmentals, right for the point of a biochemical pregnancy. Remember, this was a non-selection study, so we didn't have the answers. So these went back, there was no bias, there was no selection, and they just had higher, lower initial rates, and of course, higher loss rates, and so they produced an overall lower, they had lower initial rates and a lower sustained rate. Excuse me for misspeaking. But it was present all along. So that adverse impact on the embryo is early, and then it stays consistent. If you look at the clinical miscarriage risk, the N wasn't really big enough to find a big difference with age, and probably is not different with age. So it's, again, this causes a lot of trouble, even in our youngsters. If you look at the overall diminution of the sustained implantation rate, again, the location didn't matter. And interestingly, I think interestingly enough, the size of the segmental abnormality didn't matter. Now, if you go to the microdeletion syndromes you studied in your obstetrical rotations, you'll see that the majority are less than 1 megabase. But this is not really necessarily the same type of biology that produced those microdeletion syndromes. It may have been their genesis originally, but the biology is a little different here. Even the ones that we considered small, these are roughly quartiles of the embryos that we had by size of the lesions. The sustained implantation rates were 42% if it was small. It was 37 or 38% even if it was large. So what that tells us some things that are important biologically. And what we interpret that as saying is that it is likely that the cells that are abnormal are being eliminated. Because you find very few segmental abnormalities in placentas and very, very few in people, these cells are probably being eliminated. And the embryos that are successful in eliminating them are probably the ones that go on a sustained implantation. And the ones that are unsuccessful in eliminating them are the ones that account for the drop-off, the diminution in outcomes. If you want to understand how that works, I want to refer you to a paper by Singla that was published in Nature Communications now 3 or 4 years ago. It's just a spectacular paper. I think this is maybe one of the most revealing and insightful papers ever done. It was done by Magdalena Zerker group when she was at Cambridge. She's now at the Salk in Southern California. It really used the mini models to go through and demonstrate how mosaic aneuploids or segmentals can eliminate themselves and end up with normal embryos. And of course, some can't. And guess what those embryos do? They arrest. It's just what we see clinically. On the other hand, I will tell you that this paper is derived of 24 different experiments. I mean, really different. You could almost make it 24 papers to create this pathway that undergoes cell death for the abnormals. Aneuploids would include mosaics and segmentals and ongoing development of the cytosol. This is the absolute cure for insomnia. It took me like 5 settings to get through the paper once. I had to read it probably 5 times to truly have some even basic understanding. And Magdalena has done a lot of collaborative research. We've written several papers together with our group. So I kind of know how she thinks and writes, and it still was very painful. But if you ever really want to see how to evaluate a physiologic question, boy, there's probably not much better example than this paper, seamless paper from Nature Communications. So the nature of the segmental did not really impact the sustained implantation rates. What about in recurrent loss? We took out a little over 400 patients that had recurrent loss because we thought this might be an explanation for those, perhaps some implant longer and then go into the loss. But unfortunately, it was not true. In animal models, you can create it, and it's true, but it is not true clinically. So there were not more segmentals in the recurrent loss patients, and the segmentals had the same sustained implantation rate in the recurrent loss group as the infertile group. So unfortunately, it did not help us there. How important might this be or not be? And the reality is when you look at the distribution of results, and this is from Teague's paper, about a third or so were aneuploid. About when you use the enhanced assay, another 17% are segmental, about 4% are mosaic for whole chromosomal mosaicism, and the rest are euploid. The sustained implantation rates you saw 66, 66, 34, and 0. If you take that and you say, well, let's just look at the transferred embryos, the ones that could potentially be transferred, the euploids, and I threw the mosaics in, would make up about a little over 80%, I think 82 to 3%, while the segmentals, which some would exclude, would make up 17%. Those would all be lumped into our transfer pool now, and those would have an implantation rate of 34, and those of 66. If we could get you an overall delivery rate, which is lower, if you could make those by selecting against them and just select from this pool, all in all, you eliminate about 1 in 6 failed cycles. So your delivery rate should increase by about 6%. So 6% may not sound a lot, but if you talk to some of the other REI faculty, your colleagues that have done REI or people who just live in this world, embryologists, they would all do almost anything, 100 backflips to get their implantation rates to rise 6%. They're high enough now that 6% is an almost unattainable goal. And yet that can be done just by enhanced selection. It's not that you throw those embryos out. It's not that I would never transfer them. I would transfer them. But the long and short of all of it is that you can be more selective, get higher sustained implantation rates. Even that, however, would take us to a failure rate of about 25%. So some embryos still fail. And at least as of yet, we're probably not going to find the answers to that in genomic instability, not from aneuploidy or segmental abnormalities. And so I wanted to kind of bring you up. Some of you may have heard us talk a little bit about what we've been doing with mitochondrial changes and embryonic competence. And it's really very fascinating. The potential for mitochondria to be important was originally pointed out by David Keefe in a paper in FNS in January of '95. And since then, it's been studied, but it's become much more intensively evaluated in the last 4 or 5 years as the technologies to look at the mitochondria themselves or their DNA has improved. So this is just a graph taken from internal data at RMA New Jersey, and you can see that if you don't do PGTA, you're just selecting without taking the abnormals out, then implantation rates decline pretty dramatically with age. They do. Euploids fall even with age, even with euploids, right? So by the time you're 43 or 44, it's not bad. It's 58%, but it's also not awesome, where it should be in your 70s when you're below 30. And so the long and short of all of that is something accounts for that diminution, and of course something accounts for the failures above it. So what else can do it? We think mitochondria might be very important here. So mitochondria, everybody had this in undergraduate school and probably high school for many of you, and not where I went to high school, but where you went to high school. Mitochondrial structure, And it's a little different. They're shaped differently. They're rounder. They have different cristae formation. They're a little different inside in the mitochondria inside oocytes. But in the end, it's a double membrane bound. They have 1 to 10 copies of mitochondrion A, so that's not consistent. And they typically would have about 10,000 per cell, maybe a few fewer. They generate energy, they regulate metabolism, they're heavily involved in calcium signaling, so on and so on. So they do many important things. And we know that mitochondrial metabolism is really quite complex in oocytes. I won't bore you with what the developmental biology is. I promise you that's not going to be on your written exam in the next few weeks for the Chiefs. But the reality is it's pretty important that they be replenished and present in the mature oocyte. Mitochondria, oocytes have more mitochondria than any cell in the body, more than cardiac muscle, more than skeletal muscle. And so they obviously, energy metabolism plays a critical role in embryonic development. And there's actually quite a bit of effort made by the oocyte to produce it. However, production stops late in oocyte maturation. It certainly does not go on in early embryonic development. So you can think of those mitochondria as being batteries. Early oocyte gets so many, and the embryo has to live off that power source, off that energy source, until such time as it can start repairing them or producing more, which is really in the early post-implantation interval. Now we have some data maybe to say it begins on day 5, late day 4, in small quantities, but in wholesale, really do the job. You really don't see that until day 6 or 7. This kind of leads to our joke that you don't have to keep an embryo growing and going through development until it grows up and goes through med school residency and graduates. You really only need to get them there till day 6, because after that, the embryo will handle its own business and replenish the mitochondrial supply, not only with more mitochondria, but with mitochondria that are functional. So we think that mitochondria are really a player in early development. And these data are from Jim Tremarky, who wrote this paper now over 20 years ago. He was at Brown at the time with Keith. And the answer is unequivocally yes. There's enormous changes in oxidative phosphorylation. And there's some is energy independent, and that's pretty constant. But as the energy requirements increase, particularly after day 3, the mitochondria really have to kick into gear to provide all of that. And that's been shown by many investigators. And we know that mitochondria change with age, right? So we know they impair. We know that with progressive age, we know that there's damage that leads to protein aggregation and other problems in the mitochondria. And we know that they have a number of activators, a number of mechanisms. I'm happy to discuss these. But the main thing to think about here is that they don't undergo recycling and fusion as they should. So what happens to mitochondria, right, is you've got all this energy stuff there. You've got all this huge polarization at those intermembranes. And that leads to reactive oxygen species formation. So you can think of it like hydrogen peroxide with some organic thing stuck on the end, or some R stuck on the end. There's many different kinds of organic peroxides. And what do? They go around and oxidize stuff. They break stuff down. Part of what they break down is the mitochondria itself. It attacks itself. It oxidizes itself. And when that happens, that intermembrane depolarizes, and it moves into, functionally moves into a pool, whether it's either going to undergo autophagy, which we call mitophagy or mitophagies. People pronounce it differently depending on where you're from. It breaks down, although sometimes it can be repaired. And there are specific repair mechanisms in cells that allow it to recover and then be recircled back into the functional pool. This does not happen correctly in many oocytes and embryos, particularly embryos. So what happens is that you don't move into the ones that are just too broken to fix. They don't move into this pool that gets recycled. They just sit there. And so you see accumulation of enormous number of mitochondria inside these cells, but they're not recycling. And in some sense, they're in the way because they're still full of receptors, they're still full of calcium. So instead of having 10,000 mitochondria or 5,000 mitochondria, you might have hundreds of thousands, tens of thousands, because this accumulation, this problem has been going on for a while, not just in the embryo. And so you see, Carol Werner from our group showed, oh my goodness, back in 97, that there were some occasional embryos that had as many as a couple 100,000 mitochondria. Normal would be more like 30 or 40,000, but that's still exceedingly high. And those ones that don't work, still, when something calcium is released, they absorb part of it. And when other signals move in and out, they absorb part of that signal. And so they are there like little antagonists. And they are blocking the activity of the mitochondria and the things that are tried to do it. Embryo, who's part of our group, has looked a lot at the genes involved in this. I took those slides out in an effort to be merciful on your graduation day. But quite frankly, we've done knock-ins and knockouts, and you can certainly, selective knockouts, you can certainly alter this process by altering the things that regulate this movement from when it's broken down, it's depolarized, you block this movement into autophagy. Now, when that happens, and what happens? The cell doesn't undergo apoptosis, but it functionally can't grow. And so what does it do? It sits there and it arrests in development. Now there's an autophagy promoting or tracking gene, 5+ that's involved in making sure that autophagy, mitophagy, whatever you want to call it, occurs correctly. And when it works, embryos grow and you get babies. And this particular project was done in mice. But basically, when you can move depolarized mitochondria into autophagy, you get babies. But when you can't, you block it, you get, depending how complete the block is, you get embryos that are born, but they don't survive. If you babies that are born, they don't survive. If you really block it completely and you knock out some of the reserve and other factors that can help accelerate autophagy, then it's just embryonic lethal. You have to be able to get rid of these bad mitochondria to get viable offspring. And so this is just an example of one project, again, that was done by Tian Rang, one of our PhDs, for a while before she went back to China, in Embryo. And the long and short of all of that is that you require all of these things to be functional, and the mitochondrial copy number gets out of control when they're broken. So Peter Ferguli, who worked with our group in our UK lab, in collaboration with Dagan Wells, but also many other distinguished investigators, Jacques Cohen and others, and really thought she had found that if you simply looked at the number of mitochondria, when it was high, that indicated dysfunction, and it was potentially another diagnostic. But our control group was very small, and it turned out when we did a much larger study, 600 patients, what we found is that is not true. It's not that they tried to estimate the number of mitochondria by the mitochondrial DNA content. It's just not specific enough. So it turned out not to be helpful. And even in the days when we did 2 embryo transfers many years ago now, the one that had more mitochondria, if you had embryos that were different, was just as likely to implant and deliver as the one with low. So quantitating mitochondria is not enough. So where does that leave us? It doesn't change the fact that if you have abnormal mitophagy, if the cells, the mitochondria in the cell fail to autophagize, then you're going to have trouble. And so we have recently done something that's, I think, really fun, that'll be presented, I think, at the ASRM, where we took something called carbonyl cyanide 3-chlorophenylhydrazone. It took me like 3 tries to learn how to say that. But CCCP, which, by the way, for those of you who are old enough, is how the Soviet Union used to abbreviate themselves. That's how you remember it. And the bottom line is it encourages autophagy. It encourages mitophagy in this case. Now, you're thinking, wow, why would you want to do that? Why would you want to create more? Because you're trying to encourage mitophagy in those mitochondria that are already broken down, that are already not functioning correctly, and they need to go through this process of being taken up by an autophagosome and broken down. And so when we did that, and these are some pilot data, this is an ongoing project, we took 41 embryos from 34 patients, that had been a 2-cell on day 2 and were still a 2-cell on day 3. For those of you who've spent some time around embryology labs, a few of us have, what you find is that if an embryo is 2 cells on 2 consecutive days, that's it. It never divides again. You can say with 99% certainty there's enormous data. It dates all the way back to Lucinda Beek's data in 1986 that she presented at the World Congress. There's just no further division. And what we found is when we took triploides, our normal fertilized that had just arrested, that a substantial number resumed division. 2/3 of the triploides resumed division after they'd rested, but only 21% of the normal fertilized. That's probably because these are the more less healthy, the sicker embryos. But still, to get 1 in 5, and we did no dose response study. In fact, you can increase that number up to closer to 50% simply by having an increased dose effect on the CCCP put in. And so this is important for a couple of reasons. It demonstrates, back to this. It demonstrates that abnormal mitophagy, and these are except for the 3PNs, these normal fertilized were otherwise euploid, by the way. So it demonstrates that otherwise euploid embryos sometimes just don't have the capability to complete their process. Their mitochondria are inadequate. They back up. They're not cleared. It interferes with development. And this is an early development. We haven't done this study for day 3 to blast or to hatching blast or beyond. We actually have the capability to culture embryos to day 15 in the lab now and get to much further into embryonic development past gastrulation. But at any rate, the bottom line is that this is a process which can be altered. A CCCP is membrane soluble. So you don't have to inject it. You just have to put it in the media. And I'm not saying it's going to be CCCP, but it really speaks to the fact that if there are things that we may be able to do to enhance this process that may reduce the arrest risk. If you were to ask what percentage of embryos arrest between the blastocyst stage, between day 3 and the blast stage, without doing any genetic screen, just across the board, it's 48%. It's a lot. What would happen if we could increase that number of embryos? What if we could increase it 20%? That impact on patients would be massive. Now, you should be safe. There are certainly risks to turning mitochondria on up too high. There's a great deal of work to be done here. Please, this is not going to be in the clinic tomorrow. But it shows that there are some non-genomic instability related reasons for embryo arrest, that those are very prevalent, and that we do have the ability to enhance that physiologic process to try to recorrect them, to return homeostasis back to the embryo. At least that's the goal. I'm going to close here in my last few minutes by just talking about a bad study. So this is the day when we're doing research, right? So it's always fun to talk about what's new and what's good. Let's talk about what's bad. And so this is a paper from Yan that was published in the New England Journal. And they did a study where they wanted to see if PGTA embryos really were more often reproductively competent than not. So they did a trial where they selected 3 embryos based on morphology. And then they did PGTA, they randomized to PGTA or not. Now, if you think about that, there's 3 dots for failing. Those are 3 embryos because it's a cumulative live birth rate, cumulative live birth rate study. That's a typo. If they're all 3 bad embryos, then you're going to fail no matter what. And if it's a good embryo, because PGTA doesn't change that, you may find it on the 3rd transfer, but you'll find it. Or, in 1 of the 3, 2 of the 3, or all 3 of the 3. These are all going to be in the delivery group. These are all going to fail. And nothing about selection changes that. And so when you randomize, which they did well, each group already has absolutely equal number of failures and successes. If you're going to just keep transferring to either one out of those 3, of course, all the other embryos are there, they didn't test, or use or get a delivery, then both groups are destined to be identical. Now, that's true in the control group for sure, because you're not doing anything. That's their true state of potential. But what about in the PGTA group? Well, it can't be better. That's mathematically impossible. But it could be worse. if the biopsy hurt the embryo, or if you discarded bad embryos from overdiagnosing abnormal. It could be worse, but it can't be better. So if you do a non-inferiority study, this group can never be inferior. So it doesn't meet the criteria for non-inferiority study. Dr. Nath can beat me up later, but that's true. And if you look overall, they also didn't determine in advance, they didn't do a non-selection study to determine whether their mosaics had reproductive potential or not. They just threw them out. And of the potentially available transferable pool, it was 40%. So 40% of the embryos they threw out were mosaic, and 40% of the ones that they called abnormal, which means they threw out a ton of normal embryos. So I'm going to just say briefly, if you go, I'm not going to go through all of this, if you go through and put them all back in, put the mosaics back in and look at the delivery, you get 496 deliveries, which was within one of what the control group had. It's identical. So, but they took them out. So the reason the outcomes were slightly lower in the PGTA group is because they threw out normal embryos. So if their conclusions were conventional IVF resulted, and I apologize by not doing the study, that's in the way, I'll move it out of the way. Continuous IVF resulted in a live birth rate that was non-inferior. It was not even possible to evaluate that. It was preordained because they only used 3 embryos as opposed to screening all of them. If you look at their second conclusion, live birth rate after conventional was not only non-inferior, but was numerically higher. It wasn't statistically higher, but it was numerically higher. It wasn't possible to evaluate because the two groups weren't treated equally. And then if you look at their third conclusion, the frequency of pregnancy loss was not different. It could not be different in a cumulative sense because you transferred all embryos. So the bottom line is that this study was so poorly designed that it was not capable of not even asking one question. This was published in the New England Journal. I was one of 7 reviewers. I know at least one other one who recommended rejection. But the reality, even in one of our most prestigious clinical journals, this utter trash was published. So take what you've learned during your research. Take what you've learned in your experimental design discussions and methodology, and be rigorous. And just because it's published in a good place doesn't mean it's not trash. I'm going to close by thanking the team at the FEC who helped with all the work that you saw, particularly Shitao. She did all the horsepower on creating that 66,000 amplicon assay. She is a true genius. And then my last slide, I thought, Dr. Sauer said it reminded him of the day he graduated from residency. That's a picture of one of my colleagues took of my wife and on the day I graduated from residency. Boy, do I wish I still looked like that. But the reality is this is the day you won't forget. I still remember mine as well. And I hope you guys just have a great day. Thank you.
Thank you so much, Dr. Campbell. Thank you so much. And I just wanted to take this moment, since I'm standing at a microphone, to thank you for everything you've done for our residency program and for our residents, and even for our students who are pursuing OBGYN since I first started working with you back in 2004. So thank you so much. Are there any questions?
So much to challenge me. Don't let me off the hook.
We have one person coming up to the microphone.
I understand.
Again, thank you, Richard. That was a great talk, as it always is. And I have a couple of real basic questions, I think, pertinent to a lot of what residents always ask me, which relates to the cost and the applicability of such high-level technology to the mainstream, the people who are actually sending these patients to us. Can you comment, number one, on the cost? And then my only other question is, because I know you've done work in this, there's an implication here that the embryo transfer always kind of works or doesn't work based on the embryo itself. Maybe comment a little bit about the importance of transfer efficiency of the operator themselves and how that also impacts on pregnancy rates. And again, thank you so much for doing this.
Thank you, Mark. Those are really important topics. Those are extremely critical questions. So Shelby Neal from our group was one of the fellows right here in New Jersey with us, all of us. And she's only 2.5 years out of fellowship. She's already the director of IVF at Duke. But So Shelby did a study in showing that was back when it was $500 an embryo. That in fact, it was cheaper because you didn't do fetal transfers and you didn't store embryos. And we actually went and looked at durations of storage, really did the math very carefully. It was actually cheaper even when it was $500 an embryo. If you had 2 or more embryos, it's cheaper. When you look at in today's world where it's greatly reduced, about $200 an embryo. In fact, it's cheaper even if you have 1. So the cost of obtaining a delivery is clearly lower when you do it than when you don't do it. So it's actually cheaper because you avoid a lot of care that's expensive you didn't need. And then finally, very quickly, transfer efficiency is incredibly important. Programs internally have data that shows that within large groups, the difference in transfer efficiency, meaning babies show up, between different physicians can vary by as much as 50%, by half. So who does the transfer and how you do that is absolutely critical. And it doesn't matter how good the embryo is if you can't do an effective transfer. And transfer efficiency is still probably, even with the most skilled hands, is probably still a source of some of our failures, even when we can't see because the uterus will contract and expel an embryo. So many important areas to look at, not just the embryo. I'm just an embryo geek, so that's what I talked about. Gary, thank you.
Thank you. Are there any other questions? Yes, please, because of the microphone, please. Unless it's short and I can repeat it, but better the microphone.
Thank you, Dr. Scott, for that talk. I'm Jessica. I'm the 3rd-year MFM fellow here. To just add on to what Dr. Sauer said, my question is about what you think the role of the mother is in failed implantation rates. I know you commented on the provider's ability to implant and the embryo itself, but I would imagine that with a lot of patients having unexplained infertility, that there would be a role in potentially the uterus and implantation failure.
I'm going to be an outlier here. So again, feel free to roll your eyes. I won't be offended or anything. But the reality is there's a great paper written by Paul Pertea. He's from Fox Hospital in France. He came and did a sabbatical with our group here. And he showed that with up to 3, one at a time, cumulatively, with 3 euploid embryos, 95% of patients delivered. In fact, if you had a 4th end, we didn't have data for that when he was there, it's over 98%. So we actually think the maternal environment is not a common cause, at least for chronic failure. In any given cycle, could it be bad? Sure. All the stuff on chronic endometritis has been largely a commercial effort to create a market that I don't believe exists. And the same things, the ERA test is absolutely worthless. It's almost a scam. Sorry, but I guess I'm amongst friends, so I can speak so plainly. So really, we don't think it's the rate limiting step on a chronic basis. In a given cycle, of course it can be, but probably not chronically very often. HLA-Kerr, for those of you who want to read some very interesting work, may in fact, as we spread around the world and our HLA-Kerr status is different, may explain maybe as much as half of that 2% failure. Anyway, thank you.
I see that Dr. Goldberg has his hand up.
Thank you for that talk, Dr. Scott. Whenever I hear you speak, I am fascinated by what you guys do, and occasionally, often, my eyes glaze over because it's way above my pay grade. But my question is, are there successful implantations in those embryos that are euploid but have those abnormalities you were discussing? And do they ever become babies, and do those babies have problems?
So this has been a dominant question in our field. So you absolutely do get it, and that's such an important question. So the long and short is this: mosaics implant almost as often. So if you have a mosaicism, even for 21, this would be dangerous. Or a mosaic for monosomy X, partial deletion of X. The reality is that if they get to implantation, everything's normal after that. There are now 2 pregnancies reported where there was mosaicism in the fetus that came from a mosaic embryo, but that's out of many tens of thousands of mosaic embryo transfers. That's probably not any different than the prevalence in the general population. Or remember, when we're not screening, we put those mosaics back anyway because we don't know, and the prevalence is extremely low. So I think you can transfer them safely. With segmental abnormalities, we don't have as much data, but again, we see that diminution in initial outcomes and a few more clinical losses. But after that, we don't see that, and there's not been a microdeletion reported in one of these embryos that's been transferred, again, out of tens of thousands now. And so I think that the clinical risk is exceedingly low. The director of our genetics lab, probably the savant for the world for all this single cell genetics, low copy number genetic stuff, Chaim Jollis, when he went through, he doesn't mind me telling the story. When he went through this with his own wife, the only embryo they had was mosaic and had a segmental abnormality. And that baby is 3 years old. He elected to transfer the embryo because if it makes it, they do well. So I do believe you can transfer these safely. Now there's a paper just last month in Human Reproduction that showed that mosaic abnormalities may be more common in losses than previously appreciated. So there's some very interesting and exciting work that needs to be done there. So I'll leave a little question mark over there on the loss risk, but overall it's not increased.
All right. Well, Dr. Scott, thank you so much for this excellent talk this morning. We're really honored. Thank you.
Thank you for allowing me to be here.
All right. So now it's time to change gears.